Thank you, Tessa, and thanks to the organizers of this meeting and all the many friends which I'm acquainted to over the last few international meetings on polio and post-polio. I'm now reporting on the results of a research which has been financed by Post-Polio International, Health International in the US. Our research aims at finding whether post-polio could result from a possible chronic viral infection remaining in the polio survivors for many years after the acute event. These are results from an Italian post-polio cohort. So it is limited to the Italian experience on this kind of patients. And this is our group, small group at this time, and we enriched the group with an American friend who is originally from Russia. I will show you his contribution later on. And the post-polio syndrome we published last year with other friends, Marianne Litoff and John McFarlane, who unfortunately could not attend this meeting, and Joan Hadley, a review on the possibility that this syndrome, important syndrome, might be the consequence of an event of a chronic viral infection, which is only a possibility. And we only found an association, not a causation of post-polio by virus infection. And I will show you the results. Now, polio survivors, how many are they in the world? Well, we put in that review a number which is close to 15 million, might be 20 or something like that. But anyway, this is a consistent and important estimate of how many people are suffering from polio in the years 2000, which is an important estimate for international groups like the World Health Organization. And this is important that our associations carry on this information, which is not small. And the age of those survivors, at least in the USA, is estimated to go to become always older. And this is just an estimate of what happens. In different countries, like India, like China, the thing might be much different. Europe may be much similar to what happens in the USA. And where we are carrying out our studies. We are carrying out the studies in Varese, which is a city in the northern part of Italy, close to Milan. And now with Google Maps, you can go and see wherever you like. And this is our hospital, our medical center of the university, and our lab exactly in that area. And this is unfortunately for you, but I will be easy probably to understand. We went through methods which are of the virological studies. Virological studies are a little bit complex, a little bit, not so much, but requires to be very, 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 very pointed to the detail of the things. So it requires lots of references and biological references, lots of controls, lot of proofs, lot of time, and lot of course, persons involved, and with good hands and good, mind and good ability to overcome sometimes difficulties in research and disappointing results, which are common in our work. So anyway, we are dealing with lots of different controls for this kind of study, which aims at making practical a method for identifying remnants or residual poliovirus into the blood or into tissues of people suffering from the post-polio syndrome or something connected to polio. And the method is based mostly on so-called molecular method, which means only genetic methods. And the genome, you know, DNA or something, is composed of nucleotides, and we can read those nucleotides at this time with modern techniques like computers. And one thing is the RNA genome of those viruses. RNA is like DNA, but more primitive as a genome, and can be translated or transcribed or copied into DNA. This DNA can be analyzed with techniques which are called PCR, polymerase chain reaction, and this makes, you know, pretty easy or relatively easy to identify tracks of DNA which can be identified by sequencing or other means. So a little bit complex, but this is the so-called genome of polioviruses. I mean, these so-called genomes are represented like bars which have numbers. Numbers report the numbers of nucleotides. Nucleotides means the bases which are present in the RNA or DNA and whose distribution is different from one organism to the other. So you have heard about sequencing DNA to understanding, you know, the problem of paternity or some other problem in other cases, etc. And we are applying that to viruses. And we, in the genome of polioviruses, are trying to amplify or detect some tracts here where the red arrows are, and some small tracts. And since our methods are not so, you know, enormously powerful, so they can only detect small pieces of this genome, just to make things a little bit simpler. But anyway, for how they can be simple, we need to build so many new reagents for detecting minute quantities, very tiny amounts of poliovirus, which is not the real amounts of poliovirus which are present in vaccines or in the acute cases, but the very tiny, which may be remnants of a very old infection like 50 years. So we had to reconstruct all the chemistry of those tests with several years of work. And this is a tricky table, which is the most precious part of our work, because we hope that with this table, many other people in the world, in the US, in Russia, in Australia, or somewhere, could repeat that tests in their own patients to make us convinced that we are right and that maybe the virus can be related to the origin of post-polio. This is an important graph for us virologists, and we made tests which may detect for each test tube one copy, just one molecule of a tiny piece of the genome of poliovirus. This says 1.6. This says 16 or whatever, and this says also less than 1. Less than 1 is impossible. But anyway, our tests, they get one molecule of virus. So this is one of the most powerful tests which are present in virology and molecular biology in general. And this is more powerful than the biological tests for poliovirus. Poliovirus is this one, poliovirus 2 in this case, and this kind of test is probably 100 times more powerful than the biological tests, which, by the way, for you who are lay in virology, is an enormous amount of virus which may be present in 1 ml. 1 ml, you know what it is, just a very tiny amount. 1 ml of virus may contain 10 million infectious doses. With our system, we can find 1 billion infectious doses in that material. So it overcomes the sensitivity of biological tests, which is important. And the other point is, what is the specificity? Specificity means what kind of enteroviruses is going to detect this kind of test. Where the letter is C, this is the species where the polioviruses are contained. And these are detected with all of our tests where the red symbol is present. And then this is another table, or summary table for specificity. Polioviruses are here, and they are hit by all of our tests. But some other of the viruses may be hit by other tests, which we use sometimes. But what we are using as procedure for detecting polioviruses in persistent infection in the post-polio patients. And we take usually blood, because blood is very easy to get. And blood is known for containing lots of leukocytes. Leukocytes, of course, you know what they are, the white cells of the blood, and they may carry some very tiny amount of virus. So we co-culture those cells with cell lines, I will show you at this time, and we amplify a little bit the small amount of virus, and then we make the molecular tests here, and then detection of viral antigens in parallel, which is a little bit cumbersome in terms of hand working, but is beautiful and convincing. So sometimes from those patients, we also take tissue biopsy, but this is only rare, and we only have a limited experience on that because people don't like to have tissue biopsy like skin or duodenum or whatever else. And so, for instance, this is a tissue biopsy from duodenal cells of a polio survivor who underwent this kind of, biopsy just because he had comorbidities, like, some problems, digestive problems, and so on. So we were culturing his or her, cells, and they were, growing, and they were tested then for the presence of virus. When we did that, we tested polio patients and even other chronic enteroviral diseases to understand whether our test, based on culturing the cells before making molecular tests or antigenic tests, which is immunological tests, is better than directly attempting to demonstrate the viral genome. And this has been, so far demonstrated statistically. When we first culture the biological sample, and then we apply molecular or immunologic, we increase sensitivity by 5 or 10-fold, which is critical and important. And in the meantime, we were, carrying out this research. We enriched our panel of reagents with lots of different reagents, which are specific for the different viruses in the group of enteroviruses, which we heard today in the story of poliomyelitis. But in red, this is poliovirus 1, 2, and 3, and we have specific reagents, and we also have specific reagents, similar reagents, coming from Russia and the United States, which are very specific reagents. And this is one of the my preferred slides, because this says how they are precise when they give as a gift their own precious, antibodies. Oh, very precise, very good. And then with those antibodies, we make immunofluorescent tests, which are very beautiful in terms of pictures we got. And this is our technician, very attentive person, and he's got a particular ability in that, and these are cases from polio. And you get, with the enterovirus antibody, if you see here, among those cells in red, you can see some signal in green. And the same you can see here, and the same you can see here. Here you don't see any green. You don't see any green here also. But since the antibodies are different, these are different enteroviruses, they don't hit the polio. They only hit other cases like diabetes or heart disease caused not by polio, but caused by other types of enteroviruses. So by the research on the polio, we set up a test which might be useful not only for detecting poliovirus in chronic virus infection, but also for detecting enteroviruses in general in other important human diseases like diabetes, like myocardiopathy, which is a byproduct which we expected a little bit, but we were not sure of. Then we concentrated on post-polio cases, and we have lots of beautiful pictures showing the green. This is a case which we called Varese 15 is the year, the number 14, etc., etc. And then with some of the, antibodies, we don't find anything, and this is expected. With the polio antibody, we found green, and with the polio 1, we find very strong green. What does it mean that? That means that person who had been infected with poliovirus, let's say 20, 30, 40 years ago, still is carrying poliovirus type 1 in his blood, in his leukocytes, which is convincing because the picture is more convincing than chemistry in general. At least it's more easy to understand. And the same is case 2 or case 3, similar pictures, and you see still the beautiful green coming out with the right antibodies to poliovirus, which is right exactly here. This is a higher magnification of the same picture here. And we applied those methods to a cohort of more or less 110 or 120 Italian people who were attending hospitals for PPS, so-called PPS. They had a diagnosis according to the international criteria. But our study may be a little bit biased towards the most severe form of post-polio or polio complications on evolution or progression. Why? Because those people were attending several different hospitals before having a diagnosis. So that means they were unsatisfied with their doctors and with the cares they could provide. So they were, going from one place to the other, and then they arrived to our place. So probably our study is biased towards that. This is a group in the Italian Association. We went for a small meeting somewhere in central Italy last year. Now, these are the hospitals from which we recruit our patients. This is in Varese. This is close by in some small place. And this is in Milan, which is a larger city. And this is Malcesine. Someone of those know that place. And this is the University of Verona, where is working Laura Bertolazzi, a colleague of us. And this is our cohort, 112 so far post-polio, a few stable polio cases, which were really expected from PHI and every intelligent person, because needs to be contrasted. Post-polio, very severe, the others not so severe. Why should both have the virus? If they both have the virus has no implication. If only the most severe have the virus and the almost healthy have no virus, maybe they have got rid of that virus. And so that interesting in our situation. The age, etc. Unfortunately, so far only 18. But one thing we got easily was the family members of those patients. Family members like husband or wife or son or whatever who was accompanying the patient. And they were, happy to give us some blood or some other sample. So we tested whether virus was present in the proband and not in the family member and so on. And this is the years we have seen similar things. And in Italy, the most recent couple of cases we've got, they are close to the year 2000. And they were possibly or probably, or we don't have evidence directly, but we suppose that they were vaccine associated. And then this is, of course, PPS diagnosis how many years after paralytic polio? In general, 50 years later, more or less. And then some cases, one is very serious, progressive polio, 7 years after vaccination, already, with a clinical diagnosis of post-polio, young guy of 28, if I'm not wrong. And so we get leukocytes, or sometimes we get some other samples. We also got urine, which is of course easy, and CSF, which is more difficult, of course, and we only do that when there is a clinical indication for that. Sometimes duodenum, sometimes for other surgeries, we get small pieces of tissue. But this is the most important, summary table of our results. The summary table says that if we analyze blood donors, these are perfectly healthy people, like in every country, and never find poliovirus. If we use pathological control, which means other people with neurological disease, like multiple sclerosis or other diseases, we never find poliovirus. And if we see the post-polio patients, we find poliovirus in a considerable number of patients, 101 out of 112 so far. That is, the green part is the most part, the highest proportion, which is virus positive. Instead, in the few cases, only 18 cases of post-polio, of stable polio, only 2 out of 18 had virus inside of their body, suggesting a possible causative role of poliovirus in PPS, or that the association is not by chance. So that's the most important part. These are well-contrasted stable polio with post-polio, and the other thing, both are well-contrasted against, healthy control or pathologic control. And the other thing, that system is a bit too sensitive on my computer, but among 51 family members of post-polio, only 2 who are still in perfect health, and they are working and doing whatever, but a couple of them we found polio positive. But a couple of them out of 51 is a very tiny number, which may mean so many things. But actually, these people are living with the polio patients and almost never get polio infection, which is an important conclusion and a good message. Today, we were talking about the types of polioviruses, type of viruses which cause polio. And type 1, type 2, type 3 are the names and the vaccines which are targeted against all of the 3 viruses, and how many we get poliovirus 1. The majority of our cases are due to poliovirus 1. Poliovirus 1 is the most aggressive of all the polioviruses in existing polioviruses. So no wonder why this is the most prevalent. Then polio 2 and then polio 3. But then we have still a gray area where we could not identify the precise type of poliovirus infecting our cases. But this is due to molecular tests. But the fact that virus activity is present in those cases is not only due to the detection of virus genome, but also is documented by the finding of viral antigens, as I already showed to you, and by the production of so-called inflammatory mediators, so-called cytokines, which are produced by infected cells. Again, I will show you some of the cells from a couple of different patients. This is directly from duodenal biopsy. You see, these are the cells of the patient. If you leave them in the culture without antibodies, without immunity, without anything, very tiny amount of virus starts growing in those cells, which is very interesting and impressive. And the same is for leukocytes. You can find out some of them positive for virus, which means this residual presence of virus not only consists of the genome, but also of the antigens. But when cells are infected with those residual virus activity, they also produce some inflammatory cytokines, especially, this is this column here, the bars here represent against the controls. A little bit of secretion is always present, even in uninfected cells, even in cells co-cultured with blood donors, leukocytes, but the amount of those cytokines which is secreted upon co-culture of the cells with polio samples is much higher, significantly higher. That may mean that these cytokines may be secreted also in vivo within muscle, within the nervous system, within other parts of the body. We don't know that, but we know that in vitro the phenomenon happens. So this says that the production of inflammatory cytokines in these cultures infected with viruses, which have been isolated from PPS patients, this suggests that cytokines may have pathogenic effects, and in fact, they are known for producing chronic inflammatory changes. And that's one important point. The other point is about the genomes. We made that study of the genome, we are making actually, this part of the, because that's difficult also for them, with the FDA, which is the Food and Drug Administration in the United States, and specifically with the poliovirus enterovirus laboratory of the Food and Drug Administration in Maryland. And we have a friend here, and the friend is this nice guy, and a little bit smiling, and he's a very peculiar person. He's that's interesting, because I didn't know him. I just called him, why don't we make a study? And he said, oh, that's very interesting. I always thought that, but never did. Okay, we will do together. And I went to the States, we were discussing, and then he accepted to collaborate. And that one peculiarity of this person is that he's a virologist, he's Russian, and he emigrated to the United States in the 90s. And he's doing the same work he was doing in Russia. So studying polioviruses. But the reason why is that probably he likes biology, virology, or whatever, but he was the son of the big director of the Poliovirus Institute of all Russia. Until Siberia, His father was directing all the institute. So he was very well acquainted with the problem of polio. He's very acquainted with Sabin because he has pictures with Sabin carrying him by hand when he was 12 or something. So we were discussing with other friends, and this is a Japanese friend of mine, and how can we do that? And we are choose to do some virome analysis, some more in-depth analysis of the virus. With this same system, we are getting, you see those kind of letters. These are the sequences. Finished? Okay. I will stop almost here. And this is a sequence from a PV1 variant from an Italian patient. You see probably yellow. Yellow is variation, that is mutation. And this is another post-polio variation with regard to the standard virus strains. And this is also another patient with variations. Okay. Conclusion. This virus, multiple portions of poliovirus are existing, and the genome portions are mutated, which is important. The movie now. Does this virus produce any effect in cell cultures? Look here. This takes 100 hours to make this movie. Look here. You see the cells are gone, and they release their small parts. The difference with the regular poliovirus is that this movie with the regular poliovirus takes 3-4 hours. With the virus taken out from the PPS patient takes 100 or more hours. So that means that mutated virus is much slower. That's why we are still alive. So anyway, I'm going to the conclusions, because this is interesting for the immunoglobulin levels. And you see patients with given immunoglobulins, this is the immunoglobulin in one of our patients, you see this part of their blood, of their serum, goes much up, twice as much as normal. And the virus titer they get against polio goes very much up, 1,000, when normally this person had 32 of titer. So the administration of immunoglobulins not only has an anti-inflammatory, but also has anti-polio activity, as far as we understand. So anyway, the message is already understood. Our test, we hope, may complement the clinical assessment of PPS, and we are talking about association of virus. We are not talking about causation. And the other good point is that the poliovirus could not be detected in family members of PPS subjects. The study suggests that the possible use of immunological or chemical antivirals might be useful for curing PPS patients. I thank John Hadley, her team, and all the PHE for the support, and our friends who provided patients, of course, all the patients.
Thank you, Dr. Antonio.
Sorry. That was